Swayam Prabha. Digital India. Educated India.
Welcome back to the second lecture of the second week. So, in the last class we started with integumentary system, and I highlighted the fact that this is one system or one organ system which all of us, irrespective of our gender, irrespective of our regionalism, irrespective of which part of the world we live, we all use it. Every day we comb our hair, we ensure our nails are being cut properly, we do manicure, we do pedicure, we take care of our skin. So, yesterday when I finished the first class of the second week, I gave you the cross section of the skin. If you remember, just to have a small recollection what we talked. So, we talked about the two major layer, right, the epidermis and the dermis layer, and underneath the dermis layer is something called a hypodermis layer. Okay. So, let us put it instead of going back to the, so let us put it here. So, this is lecture 2 of week 2. Okay. So, we will be continuing with integumentary system. That is where we are, just to have a recap. Okay. Integumentary system, and today we will be talking more or less about the skin. Of course, we will talk a little bit about the hair in later half, and we will talk about the nails. Okay. So, today our thrust area is the skin. So, in the previous class about the skin cytoarchitecture, we talked about two different layers. Okay. There is this epidermis layer, then underneath is, so you remember the structure, there was convoluted convolutions and undulations out here, and followed by the dermis layer, and underneath the dermis layer where you are having a lot of blood vessels and fat tissues and everything, we call it the hypodermis layer. And if you remember, in the hypodermis or subcutaneous layer, so I give you an example that if you have to give a subcutaneous injection, then the syringe has to come all the way down, and within the dermis layer there are two parts. One is called the papillary layer, and the lower half is called the reticular layer. I think in the yesterday's or the previous class I forgot to mention the name reticular layer. Just cross check it in case I have missed out, then add this. Okay. Reticular layer. So, this is the overall architecture, how you can remember it. Now, within the epidermis, you have seen the one is the top part, this is the exposed part of the epidermis, and the other part which is underneath, as you could see the undulations which you see is the underneath part. So, this whole epidermis what you see out here. So, there are parts of the body. Now, think of it, what is the difference from this side and this side? The first difference what we talked in the last class is the color. This side is much more brighter, this side is much more darker. Apart from it, there is something more. This side has no hair, this side has hair, right? Did it strike you? They are all skin, right? So, it is basically a skin which is continuing. How come certain parts does not have any hair, the other part has hair? Why? Some of us are dark skins, whereas other ones are as fair as this or even more fairer than that. what determines. Now, today we will talk about this layer called epidermis. So, epidermis is more or less a 5-layered structure, 5 cellular layer, and they have different names. But before I come to this layer of these 5 different layers, let me give you some basic understanding or framework on which you can develop your knowledge. So, if there are 5 layers, so layer 1, layer 2, layer 3, layer 4 and layer 5. And if you notice the 5th layer, as we will draw, if you see here, it is the layer which is the lowermost layer where I have put the arrow, right. You see this one? This is the lowermost layer. Now, let us add up a bit of our knowledge. This is layer 1 from the bottom. Okay. So, I am going up like this, layer 2, layer 3, layer 4 and the uppermost and exposed layer is called the layer 5. And the way we will be studying it is from the lowermost layer, the layer which is bordering with the dermis, where you have epidermal ridges. You remember we talked about the epidermal ridges and dermal papillae yesterday, previous class. So, these are those epidermal ridges, and here you see the dermal papillae. Okay. The reason why we will be starting from the bottom, it is a bottom to up approach. The reason is there is a movement of cell which takes place from the lowermost layer all the way to up, and all the replacement what you see on the top, whatever over a period of time, these cells, skin spills out and it gets replaced by, or there is an injury, everything gets replaced. So, it happens because in the lowermost layer of the epidermis, you have a set of something what we call as skin stem cells, epidermal stem cells which are present there. These stem cells continuously divide and ensure that the uppermost layer does its function and it is removed from the body and the lower layer takes up. So, because of this from lower to upper movement of the cells, so we will discuss this topic from bottom to up. There is one more thing, whenever we talk about that something is moving from say bottom to up and likewise, and this brings us a very interesting thing, what is the rate of movement? It means this whole process is a time dependent phenomena, right, because there has to be this much it will move, this much it will move likewise. So, we will talk about that time, it is generally 3-4 weeks time it takes in the go up, and the uppermost layer what you see, the exposed layer, it remains there for around 2 weeks before it gets replaced by the lowermost layer. So, with this background I will now name those 1, 2, 3, 4, 5 what you see out here, all what I have. So, in the next slide what we will do, we will name all these layers what they stand for. Okay. So, lowermost layer, so if we call it 1, so that lowermost layer stand for striatum, and that will be that will be common, striatum. So, this spelling be very careful, germinativum. This is the first very first layer, and underneath you see this kind of epidermal ridges and dermal papillae, and it is attached. This is also called the basement membrane. It is attached to the basement membrane. We will come to that. The layer, so if this is 1, let us use another color to indicate layer 2, that is striatum. It will remain common, striatum spinosum. Okay. whenever we talk spinosome, it means it has had a spinous structure. Okay. Next, third layer, which is called granulosome. Granulosome. Whenever we use this word granule, it means there are some kind of granules which are present there. This is how you can remember spine, some kind of spiny structure. Okay. So, this is the third layer. Next, let us move on to the fourth layer. Fourth layer is called stratum lucidum, and fifth and the final layer, which is the topmost layer, which is called corneum. Now, what we will do now, we will discuss each one of these layers in detail, which is essential for you people. So, the lowermost layer, which form the epidermal ridges, I have already mentioned to you, and it increases the surface area by forming those ridges. Okay. And the cells out here, all these cells which are present here, they anchor to each other by a hook like projections, which is called hemidesmosomes. Okay. Hemidesmosomes anchors the cells to the basement membrane. Okay. Basement membrane. Now, this layer has different cell types. The different cell types which are present there, one is called basal cells. These basal cells are nothing but those cells what we call as stem cells. Okay. And these helps in replacing the keratinocytes, which are the skin cells, keratinocytes. Okay. The second cell type in this layer is called Merkel cells. This is very important. These Merkel cells are the cells which are present. If you ever forget, Merkel cells are present in this layer, in this fifth layer of these kind of skins. What does that signify? It is not present here. That signifies the Merkel cells are the ones which are present in those skin surfaces where there is no hair. Okay. So, this is how you have to remember, the Merkel cells are those specialized cells which are present there, and these Merkel cells are very sensitive to touch. Okay. They are very sensitive to touch, and they secrete chemicals, or you can call this neurotransmitters or chemicals like stuff, which stimulate the sensory nerves. So, these Merkel cells are very interesting kind of cells, epithelial as well as neuronal origin. The third cell type, I will use a different color here purposefully. So, I talked about the basal cell 1, I talked about the Merkel cell 2. Now, let me talk about the third cell type of this layer, which gives the brown tones of the skin. In other words, the pigment cell or melanocytes. It is these melanocytes which gives all that dark coloration what is there in the skin, okay, or these are the pigment cells. For the time being, this is what you have to understand about stratum germinativum. Now, next we will go to spinosome, okay, which is the spinous layer or spiny layer, which I have already mentioned. Okay. Spiny layer. and this layer, these cells, just like here they are attached to hemidesmosomes, here these are attached to each other by spiny structure called desmosomes. Okay. And these cells contains a unique cell type which is called cells of Langerhans, cells of Langerhans, and this will come later in the digestive system, we will talk about. These Langerhans cells are the one which leads to immune reaction against, they elicit immune reaction against microbes and prevent the skin from superficial skin cancer. These Langerhans cells are very interesting. They are also will come, we will talk about islet of Langerhans in the pancreas. Okay. We will talk later about this, but these Langerhans cells are present on the, from the lower side, the second layer, the spinosum, stratum spinosum surface, and they act as immune cells of the skin. Okay. So, this is what offers, if you remember the last class we talked about, it offers protection from microbes and all those things, and I told you we will talk individually the how they offer these kind of protections. This is how they offer these kind of protections. Next layer, which is called the granulosome layer. As the name indicates, it has lot of granules in it. It is a grainy layer, you can call it a like grains of a photograph, it is a grains or grainy layer. Now, this grainy layer is very interesting. So, keratinocytes from the spinous layer, let me go to the next slides talking about. So, keratinocytes moving from the spiny layer and differentiate stage where they begin to form large amount of protein keratin. And so, what is happening is that from the spiny layer, these keratinocytes, so I told you from the lowermost layer the keratinocytes or these epidermal origin cells, they are moving. Okay. So, once they move to the next layer, which is the spinous layer, from there they attain a stage of differentiation when their concentration of keratin molecules, which is a protein, keeps on increasing, and that is where they started to getting all those pigmentations. So, let me put it down for you guys and then you will be understanding. Okay. In the granulosa layer, keratinocytes moving from spiny layer, and differentiate to a stage where they, this is very interesting, as I'll, let me just finish this writing for your understanding, as they begin to form large amount of protein which is called keratin. Okay. Large amount of protein keratin, and as keratin fiber develop, so these keratin fibers develop, and as they keep on developing inside the cells becomes thinner and flatter. Okay. Cells become thinner and flatter, and there is a modification which takes place, and these cells become less permeable to water. Okay. Now, these cells then convert into this keratin convert into keratohyalin, and keratohyalin form dense granular structure, keratohyalin granules. Now, you see from where this layer got its name, and as it becomes dense, the cytoplasm of these cells, because of the presence of this keratohyalin, cytoplasm gets dehydrated minus water. Okay. They get completely dehydrated, and there are lot of cross linking of keratohyalin fibers takes place, and eventually what happens, these cells in this layer forms a huge meshwork like this, which does not allow water to percolate down. It kind of creates a barrier. So, granulosa layer, what you see is a layer. So, if you remember in the previous class, we talked about that the skin prevents the water to really penetrate very easily, and likewise so forth. So, this is how it does. There is this layer keratinocyte or granulosa layer, where it form very dark coating all over the place. Okay. So, now what we will do, we will move on to the we will move on to the next layer, which is the lucidum layer. So, we are done with this layer and this layer. Now, we are moving on to the lucidum. Okay. Lucidum layer. So, the lucidum layer is pretty much a pretty upper layer, and this layer is a very clear layer, almost transparent I am saying, not transparent, and cells are only filled with keratin. Okay. And here now, since I am now giving you a bit of an idea, the days it takes for the cells to move from here to all the way up is around 15 to 30 days. Okay. And this layer, as we will moving to the next layer, which is the corneum layer, here mostly there are a lot of dead cells, and these cells last for 2 weeks. So, if this is 2 to 4 weeks, which is 15 to 30 days, in the top it lasts for 15 to around 15 days, and the movement is like this, and that is why I am coming from the bottom. Okay. So, now this is the lucidum layer, followed by the uppermost layer, which is the corneum layer. This is the final exposed surface, contains a lot of keratinized cells or cells which are rich in keratin protein, keratinized cells. And keratinization is a process. For those who love definition, keratinization is a process of formation of protective superficial layer of cornified cells. Okay. Which are filled with keratin. So, this is the topmost layer what we are talking about. So, overall if you look at it, let me just again go back to the first picture which I drew. So, overall if you look at it from the bottom all the way to the top, there is something very unique. If these are the water molecules outside, it creates a lot of obstruction for the water to get in. But then too, you observe something very interesting. If you dip your hand or your leg in water for a while, you see the skin become slightly more like, it looks like as if they are much more glowing, and it becomes much more clean and much more slightly inflated. That happens because the movement of water is a function of what kind of water where you are putting your skin. If you put it in a fresh water, then there will be a movement of the water from here all the way to the inside. Some degree of movement will take place, and which will kind of enlarge these cells. Whereas, if you are in a sea water conditions, then sea water being very rich or very high concentration of sodium chloride, which is hypertonic solution, the fresh water from the body will move in the reverse direction. So, the direction here will be just the opposite, and what will you observe is your body will get dehydrated eventually. That is why, suppose somebody, there is a shipwreck or something, somebody is in kind of swimming in the ocean. This is always a problem because your body continuously gets dehydrated because water moves from inside to outside, because sea water is very high concentration of salt and it pulls the water, fresh water from your body outside, because this is one of the major problems. Whereas, on the contrary, if you are in a fresh water, then part of the water gets into your system. Of course, that enlarges the outer layer a little bit. Okay. So, keep these things in mind, and as we have already talked about, there are layers which prevent microbe infestations, there are layer which prevents water to kind of restrict the movement of water. Like I won't I would say they do not prefer the water molecules really to move through, but like kind of they prevent that it should not be that your body or the cells get inflated just because you are in water. Okay. So, there are always a restriction in the movement of the water molecules. So, overall what we see is skin is a very, very dynamic structure, and underneath this layer. So, what we will be dealing in the next class, we will talk about the dermal layer, where you have lot of blood vessels, enriching the blood vessels along with the adipocyte tissues and everything, and how these regulates different activities. But even at the top 5 layers, there are very, very interesting features to deal with. Next, what we will do after this, once we are done with this part, we will be dealing with 2 interesting topics. One will be the skin color, and the second one will be the function of the dermis and some of the functions out here, and then of course, we will talk about the cytoarchitecture of the hair and the nails. So, these are the tail pieces which are left of the integumentary system that we will be dealing in the next class. Okay. Thank you.